Good morning, everyone. Let's get started. Welcome to Grand Rounds. My name is Adit Talati, and I'm one of the Grand Round directors, along with Drs. Rebecca Muley and Alex Harris. Today, Dr. Alex Harris is online. You could see him. And for those who are online, you could post your questions in the Q&A panel, and then he will curate them and feed them to us so we could read them out for Dr. Gur. You don't have to wait till the end. You could post them during the talk as well. Also, some news. The next two Grand Rounds will both be in person. We're very fortunate. Next week is the Mumford Grand Rounds presented by Dr. Mary McKay, the Executive Vice Provost at Washington University. The title of her talk is It Takes a Village, Collaborative Partnerships to Advancing Child Physical and Behavioral Health. The week after that, April 9th, is our own Jean Marie Alves Bradford, Professor of Psychiatry and Vice Dean for Student Affairs. Her talk is an interactive one, and it's entitled Improving Learning Environments Through Upstander Skills. So we look forward to seeing you there. Today is also a very special lecture. It's our Women in Science Empowerment series. I'd like to thus invite Dr. Stephanie Collins-Reed and Wei Li Chang to our department as they'll introduce the WISE series, why it's so important, and then further introduce today's speaker. So without further ado.
Thank you, Adi. So I'm just going to take a minute to talk about WISE before we introduce our speaker. WISE stands for Women in Sciences Empowerment, and our mission is to empower our women and non-binary faculty colleagues with skills for career and personal advancement, and by creation of a tight-knit community that support and advocate for each other. I, along with my co-chairs, Wei Li and Danny Dimitriou, who is hopefully joining us virtually, she's in clinical service at the nursery today, would like to thank all of you who have supported our efforts in overseeing the WISE initiative over the past few years. And as always, we invite all of our colleagues to join us for future events. We encourage you to become involved in any way that works for you, and please reach out to us if we in turn can support you in any way. So thank you, and with that, I'll turn it over to Wei Li.
Great. Hello, everyone. So Dr. Tamar Gur, our speaker today, received her Bachelor of Science in Biology from Brown University and then completed her MD and PhD as an MSTP student at the University of Pennsylvania. Her doctoral work centered on the transcription factor CREB and its role in neurogenesis in response to antidepressant treatment under the mentorship of Dr. Julie Blendy. Dr. Gur then completed her residency training in psychiatry at the Hospital of the University of Pennsylvania. And as a research track resident, she worked with Dr. Neil Epperson, who was actually last year's WISE Grand Round speaker, on topics in reproductive psychiatry. She then started a faculty position at the Ohio State University College of Medicine, where she is now an associate professor with tenure in the departments of psychiatry, neuroscience, and obstetrics and gynecology. She is also the inaugural endowed director of the Soder Women's Health Research Program, where she directs efforts to investigate women's health across the lifespan with a focus on stress. Her clinical work focuses on treatment of perinatal depression and anxiety, and her translational research laboratory focuses on the link between prenatal stress, microbiota, and inflammation. She uses a variety of techniques, including rodent models and clinical cohorts, to investigate how stress during pregnancy shapes the developing brain. She also has been involved in education and is the associate director of the MST program at Ohio State. She has been a champion of women's mental health and women in mental health and a role model for physician scientists. We are sorry that we could not bring her here in person today, but we're delighted to have her here as our third annual Women in Sciences Empowerment Grand Rounds invited speaker. So please join me in welcoming Dr. Gur.
Thank you so much. Only person sorry that I'm not there is me. I would have loved to have been around so many. It actually feels like a family and friends at Columbia. And you are all top of mind, and I'm just delighted to be in community with you, even if it is virtual. And thank you to the organizers, to Adi and Alexander for inviting me, and to Danny and the other WISE organizers. And they promised they would send me the plaque in the mail, so let me know when I can expect it. I'm really excited. I'm really honored. Neil's been an incredible mentor, and Lisa Monteggia also the first recipient of this award. So it just means the world to me, and I'm really appreciative. So my talk for you today is really about, as a psychiatrist, but also a basic scientist, I think one of my North Stars has been to focus on work that really to harness the mouse to try to do things that we can't do in people for a variety of reasons, but never to forget that really my ultimate goal is to help women, right? And to help the developing baby. So not to get so enmeshed and admired in specific proteins or transcription factors that I'm not always thinking about, but how can we help women and babies with this? And so that's really going to be the defining, the through line in my talk today. And so really my disclosures, I do receive compensation from Biological Psychiatry and the Society of Biological Society for my role as social media editor, though I am stepping down. So if anyone's interested in that, feel free to send me an email. I'm happy to tell you more about that role. I think the application for the next one is open. It's been a wonderful way to meet actually many of you. I met many of you by interviewing you for the podcast for Biological Psychiatry. I used to be taught before I went to medical school, but not that long, that pregnancy was actually a protected time, that women were thought to sort of float through pregnancy on a cloud of pink pheromones, just excited for their upcoming bundle of joy. But increasingly, we recognize that actually pregnancy is not a protected time period in terms of mental health. It's actually a time where a woman is robbed of joy about a transition, and up to 6 to 23% of women, depending on the report, actually do meet criteria for major depressive disorder or an anxiety disorder during pregnancy, which is about the rate we see outside of pregnancy as well. So not only is pregnancy not a protected time, it's actually a time where mental health issues in the mother can actually be impacting the developing child as well. And so we know now that stress is a risk factor for psychiatric disorders in general. Times of transition, like going off to college, retirement, getting married, things like that, even joyful things like getting married, times of transition, and certainly becoming a parent or inviting a new child into your home is certainly a ginormous transition. And so we know that that's associated with heightened risks for mental disorders, and during pregnancy, it's for the next generation as well. And so, when I'm talking about stress, I'm talking about major things like the fall of the Twin Towers, but I'm also talking about things like daily hassles, which is what this cartoon is meant to illustrate, the hassles of daily living. So it doesn't have to be a catastrophic event, even just the hassles of daily living, and I have some citations for you down there at the bottom, have been shown to increase risk of psychiatric disorders in the mother as well as in the next generation. And so really our psychiatry as a field has increasingly acknowledged that in these overlapping concentric circles of stress and mental illness, really inflammation has emerged as where these two factors meet. So inflammation is now increasingly recognized. The immune system is increasingly recognized as a contributor to many psychiatric disorders, including depression, anxiety, and schizophrenia. And so really the big picture for my talk today, and this is literally a ginormous picture of a pregnant person, there's been increasing attention to how these first 9 months do shape the rest of your life. But really the big picture take-home message, if you remember one thing from my talk today, is that exposure to adversity during gestation is a risk factor for disorders such as schizophrenia, depression, and anxiety in that next generation. And really what I and many people, including at Columbia, are interested in is how do these events that happen in utero contribute to these long-term behavioral changes or psychiatric disorders. And this is really building upon the work of a British cardiologist, Dr. Barker. During the 40s, he noticed in England following World War II, infants born to mothers who experienced World War II and the stress of World War II, the bombing by the Germans in London and other parts of England, those infants grew up to have increased risks of cardiac events. He was a cardiologist. He was interested in cardiac events. They had increased rates of stroke, heart attack, as well as obesity. And so really this idea, he shaped this idea that many of us are following developmental origins of disease. Doe had for short that there is an in utero start to many disorders that we see in adulthood. And so that's really sort of the big picture, bird's eye view of what I'm interested in. So that's what this is. What this is not is the refrigerator mother, which is actually, I think it's important to acknowledge that in psychiatry, we have a long history of blaming mothers. If it was the 1950s and you were a woman who brought in your child for evaluation for autism spectrum disorder or schizophrenia, oftentimes they were told that it was their fault that they were a cold mother. So this is a refrigerator asking, how was school, dear? So I'm just here to be very explicit and very direct that in no way do I blame a mother for being stressed during pregnancy. That's a ridiculous idea. I think that I'm just here to, if anything, say we have to support women during pregnancy and help women during pregnancy, because not only are we helping them, we are indeed helping this next generation. But I just want to be very explicit in my conversations with my patients and my scientific conversations with colleagues that in no way am I saying to a woman, why are you stressed? Calm down. We all know that saying calm down to someone under stress. I hope we all know that saying calm down to someone under stress is, in fact, generates oftentimes the opposite result. They will not calm down. So when we think about what my topic, which is the microbiome, this is part of a larger movement in the field. And I was lucky enough and delighted to guest edit a special issue of Biological Psychiatry this last year with Elaine Sau, who's really a force in this field, about the exposome. So the exposome is no less than the sum of all the things we encounter during our lifetimes. So this is things such as stress, as I'm going to be focusing on today, but pollution, our diets, medication. And so if you're interested in this topic, please pick up this issue of Biological Psychiatry. This offers articles across the lifespan, but also across psychiatric disorders. So there's very likely that there's something of interest to you in that special issue. And really thinking about the microbiome as that interface between, and I can't believe a leaf blower is walking by, could more things go wrong? Yes, is the short answer. But the microbiome, I really think of it as the interface between the world out here and increasing risk for psychiatric disorder. So for the purposes of my talk today, I'll be focused on pregnancy in this time period as well as stress. And so what do we think of as sources of psychosocial stress? Well, I think of these across several layers. I think of interpersonal stress, such as the sleep deprivation, whether you've been exposed to viruses, what your diet is, things of that nature. There's interpersonal stress. So is your job very stressful? How are your finances? Do you have social support in terms of your partner, in terms of your family of origin, in terms of colleagues and friends? I think those are really important. There's also structural and institutional sources of psychosocial stress that feels increasingly obvious, but things like pollution, climate change, your built environment can also be contributing. And then in general, the world as a whole and your place in it can also be a source of psychosocial stress. And so these can all have an interplay and can occur simultaneously, but this is how we conceptualize psychosocial stress. And really what, I guess another North Star for me is I might not be able to fix the world out there, but if I can pinpoint the downstream pathophysiological consequences of stress for a pregnant individual, and modify them, that's always going to be the goal, to modify them, then maybe there's a therapeutic opportunity. So yeah, I can't fix the world, but maybe I can really delve into that downstream pathophysiology. So that's another North Star. How many North Stars can someone have? I think I've already named 2. So a lot of work to date on the biological mechanisms of how does stress in pregnancy increase that risk of psychiatric disorders in the next generation. A lot of that work has been done by your wonderful colleague, Catherine Monk, and is really focused on a few different areas. And so a lot of the focus has been on epigenetic programming. So it's not just the genes you get from your parents, it's how they're modified and expressed. There's been a lot of attention paid to the immune function, the immune system in the mother, and whether inflammation is coming from, there's a whole field of maternal immune activation, thinking about how does virus or immune activation during pregnancy, how does that get translated to the developing brain? HPA axis, of course, the stress response has garnered a lot of attention, and rightfully so, in terms of there's evidence showing that an infant exposed to higher levels of depression in the mother, really wonderful work from Tim Oberlander, that the infant will have a dysregulated HPA axis for quite some time. So that's where a lot of the work has been focused. And really what I've introduced along with several other colleagues is this idea that the gut-brain axis might also be contributing as a key mechanism through which stress is transmitted from the mother to the offspring during pregnancy. And that will be the focus of my talk today. And why is this important? I hope that I'm preaching to the choir by telling you that prenatal anxiety and perinatal depression are highly prevalent. This is the global prevalence. This is important not just inherently important, it's important because it's really has a high level of morbidity and mortality in the population. So really, what I was tasked with early on in my career, while presenting this work at I think it was SOBP, someone very eloquently asked me, or not so eloquently asked me, the brain's all the way up here, the gut's all the way down here. How on earth can one be relevant for the other? And so I'm going to be spending a little bit of time introducing that concept to you right now. But really, when we think about it, there's three main ways that we think stress is transmitted. One is through vertical transmission, so at delivery. The mother bequeaths her microbes to the infant. That's a very nice way of putting it, through a standard vaginal delivery or through C-section, through modulation of the immune system. The infant's immune system has a key role in their long-term mental health and health. And we think that's one way that stress and the microbiome might be modulating outcomes. And finally, through microbial metabolites. And I'll be spending some time, so the products that the microbes produce are really biologically active. So I'll be spending a little bit of time on each of these in my talk today, and hopefully you'll walk away with a real understanding of how we think, where the field is right now in terms of thinking through these things. So stress during pregnancy increases translocation of oral, gut, and likely vaginal microbiome into the circulation of the mother. This can change circulation levels, is associated with preterm birth, and then can have a wide variety of neurodevelopmental outcomes that I've delineated for you here. So to delve into this idea in even more detail, for the purposes of my talk, I've illustrated a pregnant woman, a pregnant person, but aspects of this are true for all of us. So when stress impacts an individual, they have HPA axis activation, release of ACTH, and glucocorticoids from the adrenals, as well as sympathetic nervous system activation. This can differ if it's an acute stressor or a chronic stressor. There can be some adrenal fatigue or different. This can be modified based on whether or not this is an acute or a chronic stressor, but this is in general how we think stress impacts the individual. This can also have an immunomodulatory effect. How many of us have gotten sick after putting in a grant or after finals? This is because our immune system is modulated by this stress response. And all of these things together can go ahead and impact the gut, the intestinal epithelium, and change release of, first of all, it can change motility of the gut, but can also release the physiology of the gut and the acidity, and as well as a variety of other gut contents. And now, why is this important? Well, illustrated here, we have the gut microbiome. I should stop and say that the microbiome has several communities in your body. Every orifice has its own, sorry to get a little graphic here while you're drinking your coffee and eating your pastries, but each of your orifices has its own community structure of microbes that contribute to your health. And I'm very much focused on the gut microbiome, but I will be showing you data from the vaginal microbiome in pregnancy as well, which we also think is important. So stress actually shifts the microbes that are present in your gut. This is work from my mentor, Mike Bailey, as well as several other labs that show that stress shifts the community. So if you have, let's say, microbes A, B, and C under non-stress conditions, after sufficient levels of stress, it actually will shift it to community, let's say D, E, and F. So the microbes present in your gut will actually shift with stress. And why is this important? Well, in pregnancy, it's important for a few reasons, but I'll focus first. In general, this is important because stress impacts. This is one of those dumb moments. The microbes impact the immune system. Of course they do. So this is bidirectional modulation of the immune cells, of the immune system to the microbes, but also the microbes can increase inflammation, especially if they're more pathogenic or less beneficial microbes. They also, as I mentioned at the start of the talk, are really not just sitting there twiddling their thumbs. They're actively producing metabolites. They're the key metabolizers of fiber. This is butyrate, which is a short-chain fatty acid, which is very biologically active and has a key role in epigenetic regulation. And if we were in person, I would try to call on someone and ask them what this is. But since I'm not in person, I'm just going to go ahead and let you know that this is tryptophan. And it's a key precursor, a key amino acid that we get through our diet. And I'll be spending a lot of time later in my talk on this, but we now have evidence showing that there's a reduction in tryptophan metabolizers. And of course, one of tryptophan's most important metabolites is serotonin. And actually 95% of your body's serotonin is actually in your gut. And so I think there's clear implications for how that might impact not just your health, but your mental health. And so if there's these changes in metabolites and changes in the immune cells at delivery, this is even more critical because the mother is, as I mentioned, bequeathing her microbiome to the infant. And that's what's going to set up the infant's gut-brain axis for the rest of their life. And so there's actually a lot of evidence emerging that infants born to C-section who are colonized with more skin microbes as opposed to the more enriched diverse microbiome from the gut have increased risk of atopic disorders such as asthma and eczema, and probably likely other disorders as well, but those I'll focus on for today. And so really it is important to set up that gut-brain axis in the most healthful way possible. What my lab is focused on is microglia. So that's the resident immune cell. I'm not taking you from the immune cells just in general in the periphery to the immune cells in the brain. And so the microglia are the innate immune cells in your brain. And so we know that they are important throughout your lifespan, but in development, they're really critically important. So they have really important functions in neurodevelopment, such as pruning synapses. They phagocytose, so they prune synapses. They facilitate spine formation. They promote axon growth and fasciculation and support oligodendrocyte development and myelination, as well as, really importantly, regulate neuroprogenitor cell survival. So I think I touch on almost all psychiatric disorders that I know of when we think about things that are happening on molecular levels. And so it's easy to understand that if microglia are impacted by the microbes and the metabolites and the immune modulation, that this could really alter neurodevelopment. So I hope I've convinced you of that in these slides. And so we actually had my colleagues and I had a paper in that special issue I mentioned that really illustrates some of this point. So this is a very complex slide. What I'm really trying to point out to you is that a lot of the things you might care about in your own research could be potentially impacted by prenatal stress through the microbiome. And so if you're interested in any of the things listed here on the slide, please do check out our review. Love it if you'd cite it. And so prenatal stress is leading to the stress-induced microbiome-dependent effects. And so this is what I'll be focusing on for the first part of my talk, is the shift in maternal gut microbes, and then the stress-induced and microbe-induced changes in chemokines and cytokines, and how I believe work from my lab has pointed to this really impacting long-term behavioral changes. So first, I'll introduce the model, the preclinical model we use in my lab of prenatal stress. This is a restraint stress done between gestational day 10 and 16. It's probably a pretty mild stressor, mild to moderate at most. This is not a severe stress. It's just 2 hours. The pregnant, a mouse mom is called a dam. I'm not swearing. That's just what they're called. And she's restrained between 9 and 11 in the morning, between gestational day 10 and 16, which roughly corresponds to the second trimester in humans. For those of you who are aware of the clinical literature, that is a really key, has really been implicated in perturbances during that trimester, has been implicated in increased risk for several psychiatric disorders. There's many key differences, obviously, between mouse and human pregnancies. I just want to point out that at delivery, this roughly corresponds to the third trimester. So a lot of neurodevelopment is happening ex utero or after pregnancy in the mouse. So obviously mice are not small humans, but there's a lot of differences. I also would like to point out that there's differences in the pregnancies themselves. Mice have a bicornate uterus. Each fetus is individually housed in their own little corner of the uterus with their own placenta. I'll be showing you a video in a few minutes how we use this to our advantage to manipulate the pregnancies. And obviously, there's about 8 to 10 mice per litter. In human gestation pregnancies, it's most common to have a single fetus, and there's other key differences. So I just want to just put out some of the limitations of the model right up front. So we do the stress, and then one cohort, we do placental and fetal brain tissue collection the day after the stress is over, though I will be showing you some data from gestational day 16, immediately after the cessation of stress. And other cohorts of mice we allowed to go through parturition and into adulthood. This is not a 2-hit model. There's not an additional stressor. The only stress a rodent is experiencing is in utero. And then we do behavioral testing and take other tissues here on postnatal day 70. And so first I'll be showing you microbiome data. So this is a PCOA plot. So each dot represents the whole sum of the genome, microbiome genome. So each dot represents all the microbial genes. Here is the dam, here is the female offspring, here's the male offspring, and here are the citations. So the closer together two dots are, the more similar they are. The farther apart they are, the more dissimilar. Blue is control, red is stress. And what I'm showing you here is that there is a significant change in the microbiome in the dams following the stress. And then when we took the microbiome samples in adulthood, we found a significant shift in the microbiome just with this stress exposure. And again, there is no second hit of stress. There's simply just the exposure, which really speaks to this concept I spoke of a few minutes ago, which is that they're seeded with a different population of microbes at delivery, and this extends into adulthood. This really layers and layers and layers. There's this principle of ecological succession. So if your first layer of microbes is microbe A, it's more likely to give rise to B than C than D. Whereas if your first microbe is F, that's more likely to give rise to microbes G, E. Did I just forget the alphabet? F, F, G, H. There we go. So it really does change this principle of ecological succession. If you're seeded at delivery with one set of microbes, it changes your microbiome into adulthood. And so I think that's critically important, and I'll be speaking more about that in human studies as well. So their microbiome is different. I'm now shifting our attention to behavior. This work was done by my very talented first MSTP student, Helen Chen, who's now an intern. Unfortunately, not in psychiatry, but in pediatrics. I guess it's fortunate for pediatrics. I did try to convince her that this is the best field, I promise. And so what she does here was a social, three-chamber social behavior test. So in this little cubby here, there's an object. In this one here, there's a mouse from a very docile mouse strain, and we simply measure how much time does it spend approaching and socializing with a mouse. It can only sniff the mouse. It can't attack it or really physically interact with it, but it can spend time sniffing and investigating it. And what we're showing here is a heat map from one of our mice. And what we found is that the control mice in general preferred to socialize with another mouse, whereas stress, they no longer had that preference. They were just as interested in the object. So we found a significant reduction in time spent engaging in social interactions in mice exposed to prenatal stress. And this is male and females in this, and we've replicated this finding several times now. So there's a reduction in social behavior. We've expanded and extended this to other measures of social behavior, but in the interest of time, I'm showing you the three-chamber social behavior test. So I've showed you a change in microbiome, change in behavior. We also see increases in anxiety-like behavior in female mice and changes in cognitive behavior in female mice in adulthood. Now I'm changing gears a little bit and showing you some neuroinflammation. So now we're looking at the brain, of course. So we look at 2 time points. Here's fetal brain gene expression. So this is qPCR. And so IL-6 is a really key cytokine. Cytokines are I hope there's no immunologists on the call, but I think of them as the neurotransmitter of the immune system, and that they communicate between different cell types. They recruit immune cells to the site of injury or to a site of activation, and so they're very key. So IL-6 and TNF alpha are very key pro-inflammatory cytokines. There's been a lot of work done on IL-6 by other labs as well, showing that they're key in changes in IL-6 perturbations in both humans and mice are associated with poorer developed neurodevelopmental outcomes. So we do see a significant increase in IL-6 in the fetal time point. When we turn our attention to adulthood, we've done both immunohistochemistry as well as flow cytometry to look at the immune cells. And what we found coalesces to show that there's increased neuroinflammation in adulthood. So this is qPCR showing an increase in IL-1 beta in adulthood, as well as IL-6 in the cortex of adult male and female mice. And then we also did immunohistochemistry, and we labeled, this is just a representative graph, a representative picture. IBA1 labels microglia, those innate immune cells. We found a significant increase of IBA1 labeling in the prefrontal cortex, which is, of course, a brain region very implicated in many different behaviors, including social behavior. I did not have an a priori hypothesis that there would be global neuroinflammation with our prenatal stressor, and in fact, in the motor cortex, we did not see an increase in IBA1 labeling. So this is a controlled brain region, and we did not see increased neuroinflammation there, suggesting it's more specific. We recently did a deeper dive into the fetal brain, and I'm just showing you microglia, though we do have other cell types that we are preliminary single cell RNA-seq data from. So for those of you who are interested in such things, these are our clusters of fetal microglia, M0 through M5, and this is really great work from Brandon Borowski, another very talented MSTP student who unfortunately wants to be a neurologist. I think I'm 0 for 3 now for convincing MSTP students to go into psychiatry. But what he did was he separated out these fetal microglia clusters. We don't see a change in the proportion of microglia with stress, but what we do see is that prenatal stress really perturbed the inflammatory phenotype and led to a more pro-inflammatory phenotype. We had to enrich for microglia. There's not a lot of microglia. So we separated them out and enriched for them and found a increase in myeloleukocyte activation, differentiation, and immunity in single cell in microglia taken from stress in comparison to control mice. He also looked at genes implicated in schizophrenia, autism spectrum disorder, bipolar disorder, and MDD, and found an enhanced or enrichment in psychiatric disorder susceptibility genes in the database. And so this is really tantalizing data that we're continuing to follow up on, and he's still analyzing, suggesting that microglia are changed by prenatal stress to a more pro-inflammatory, enhanced psychiatric disorder susceptibility genes with prenatal stress. So we're actively following up on this, and that manuscript is in preparation. So we think that stress shifts the microbiome and leads to increased cytokine and chemokine production. Chemokines are messengers within the immune system that also serve to recruit immune cells to the site of injury. And CCL2 early on in my lab came to our attention when we removed amniotic fluid from those mouse pregnancies and ran an ELISA and found it to be significantly increased. So CCL2 has been on my radar since I practically started my lab. And so we asked the question, in the absence of CCL2, what happens? Do we still see inflammation, and do we still see behavioral changes? Is this a key part in this pathway? And so we took CCL2 knockout mice. These are constitutively knocked out. And in fact, when they went through stress, we no longer saw this increase in IL-6 or TNF alpha in fetal brains compared to control brains. So that was a really important clue that CCL2 is required for that inflammatory response following prenatal stress. Even more interestingly, Helen took those CCL2 knockout mouse and looked at their social behavior, and they no longer had that reduction in social behavior that I showed you a few minutes ago. So it really looks like we've stumbled upon a really key pathway that links prenatal stress to long-term neuroinflammation, as well as these behavioral changes. Now, Helen wasn't satisfied. She was nearing towards the end of her PhD. She wasn't satisfied just to show in knockouts and constitutive knockouts. There's limitations to that it was required. She wanted to see if it was sufficient. So if anyone's squeamish, this is your trigger warning. I'm going to be showing you a video in a moment of exactly what Helen did. But what she did do, I'll just describe it for you verbally, is she did intraamniotic injections of CCL2, recombinant CCL2, into the mouse pregnancies. I showed you earlier that they are all individually housed. So she took that opportunity to do injections in both ends of the uterus of recombinant CCL2 or saline as control. And then she collected in one cohort all of these things, placenta, maternal plasma, amniotic fluid, and so on. And then another picture, and then, she sewed them back up afterwards. And so some the next day she collected, and the others delivered just fine. And went into adulthood where we did social behavior. So I'm about to show a video. If you're squeamish, close your eyes. But this is Helen carefully injecting into one corner of the uterus. The mouse is appropriately anesthetized. And this, for the purposes of the video, she put ink in there, and you can see the ink is traveling throughout. I believe she already did the injection in this, so you can already see there's some dye there. This traveled throughout the uteri. They're connected. Recombinant CCL2. So what did she find? And this is a really wonderful approach that we're using now with several other factors to really get at this question of sufficiency to induce deficit. And so what she found, first of all, I'll show you that right here on the left, that in the maternal plasma, there wasn't an increase in CCL2. There was also not an increase in CCL2 in the placenta. So it really was limited to the fetal compartment. It did not enter the maternal compartment. But it did, we did find it 24 hours later to be significantly increased in the amniotic fluid, the fetal plasma, the fetal liver, and the fetal brain. And really, remarkably, what we found is that it was sufficient to no longer, so the saline injected mice still had a social preference and preferred to spend time socializing. Those injected with CCL2 no longer had a preference to socialize. So it was sufficient to induce changes in social behavior only in the female offspring, which I'm happy to answer questions about why I think that is the case. But as I mentioned at the start, I really was asking myself, does this translate? I didn't set out to help mice. I did set out to help women. So I wanted to get started on that, answering that question sooner rather than later. So I did that in a few ways. So this is where I'm sitting right now today. I'm sadly not with you in New York, here in Franklin County, Ohio, which is right in the center of the state. My kids like to say that means it's 4 hours from anything good. But here we are. Franklin County, unfortunately, has high levels of infant mortality and poor maternal pre-pregnancy health. It's really a critical issue here. And you can see that up in Franklin County, this is from 2022, but this has continued. It has a high rate of a depression diagnosis during pregnancy. Almost 19% of women had that. And postpartum depression is regarded as a common issue in many communities. We also have, as I mentioned, we have high levels of infant mortality, and this is reflected in high levels of preterm birth as well as low birth weight. So it's really a problem here in Franklin County, Ohio. Look, it is many parts. And COVID-19, as we know, was a really a sledgehammer to mental health with higher rates of anxiety and stress during pregnancy. So we went out to conduct a pilot study. I'm going to tell you right now, we had a terrible, not terrible, I shouldn't say that. It was a pilot study, so our sample size was only 36. But we really I just was really eager to figure out sooner rather than later whether the things I was seeing in mice I could see it in people. So I'll tell you right now, this was a clinical study designed by a preclinical scientist. So I really just wanted to get my hands on some samples. So any issues with this, please don't blame Therese or Britt Worley, pictured here. It's all my fault. I just really wanted to. This was designed again by a preclinical scientist. So these are our inclusion criteria. These were our exclusion criteria. What we lacked in N, we tried to make up for with a longitudinal nature. So we did get many studies in this space have one time point either at delivery or maybe the second trimester. We enrolled at the first trimester and tried to get longitudinal samples throughout pregnancy. Of note, probiotic intake was an exclusion criteria because our main outcome was changes in the microbiome. So let me take a moment to say that I don't think we know enough about the human maternal microbiome. I think that's still in its infancy, pardon the pun. In general, we think that the gut microbiome, greater diversity is generally seen as beneficial, and there are shifts in composition across pregnancy, but a lot of this work is really done from a space of pathogenesis. So, C. diff is a terrible microbe. And so that's really part of why we think that more diversity is beneficial, is because a lot of that work comes out of work on the pathogen C. diff. And so these are general ideas, and here are some citations for you if you're interested. In juxtaposition to the gut, the vaginal microbiome, it's actually diversity decreases across pregnancy, and a higher level of diversity in terms of the vaginal microbes is associated with an increased risk of adverse obstetrical outcomes. But we do know that over the course of pregnancy, there are shifts in these communities, with actually the vaginal microbiome coming into the gut thought to be one of the signals for delivery. And so really our question very simply, again, was just whether or not there's shifts with stress, depression, and anxiety during pregnancy, and in the same thing in the vaginal community, and whether or not this was going to impact the infant. So this was our study design. Each of these visits, we took psychometrics. I showed them for you here. So a stress scale and the CESD, and then we got some biospecimens. So the OB pictured earlier, I'm lucky he's my husband. I'm lucky for many reasons, but one was that he just did what I asked of him and just collected these samples on his patients. So his patients in the resident clinic are underinsured, and his patients in the faculty clinic are insured with Ohio State Insurance. So that created a diversity in, as I'm showing you here in our demographics, we were very intentional in trying to recruit non-white participants that have been historically excluded from research. That was very important to us. And so though our sample was small, we were able to sample from that. And our PSS scores and our CESD scores, in case you can't remember them off the top of your head, these are low, moderate, and high. So you can see that we had about a moderate level of stress as well as CESD scores in our demographics. And again, our objective was fairly simple, just to see if stress and depressive symptoms are associated with differential abundance of maternal microbial taxa. So the short answer is they are. It was actually quite, and I'm just going to go a little bit faster because I just noticed the time. We noticed that there, even though we had such a small sample size, we were able to pick up on differences as it associated with stress. And so this is now the PSS scores. We did find a significant change in lactobacillus. There was an increase in the third trimester of lactobacillus. This is contrary to expectation. Lactobacillus in general is thought to be beneficial, but of note, it is the dominant taxa of the vaginal community, especially during pregnancy. And so what we're wondering is, and we'll need to further studies to really investigate this, was there an earlier shift from these vaginal microbes into the gut, which could potentially be deleterious? When we looked at, and there is as I mentioned, an increase with natively vaginal taxa outside of lactobacillus. And there was also an increase in more pathogenic microbes that are generally found in the urogenital tract, which does support this idea that there was a migration from the vaginal microbiome community into the fecal microbiome community. And it's associated, this S. amnii is associated with gestational complications as well as intrauterine inflammation, most notably. And Gardnerella is associated with increased OB complications in more diverse vaginal microenvironments. So it does suggest, even in our small cohort, that we were able to pick up on shifts and changes in the microbiome associated with stress. So that was interesting to us and published last year in BBI Health. We next turned our attention to stress and depressive symptoms and maternal blood. And what I really couldn't believe actually was that CCL2, that chemokine that we picked up in mice, actually was what came up as significant in maternal blood as being significantly increased and associated with stress and depressive symptoms. I just couldn't believe that actually translated to humans, but it does look like that is a really important chemokine. And certainly something that we'll be continuing to follow. And so what I'm showing you here, this is third trimester blood. We did not see any relationship in the umbilical cord at delivery of note. So stress and depressive scores were associated, sorry to jump ahead, with increased maternal CCL2. So that's something we're very interested in following up on. At delivery, a lower level of the fecal lactobacillus, which is a beneficial commensal microbe, was associated with increased CCL2 in umbilical circulation. So the teasing out that relationship between the microbe and CCL2 seems like it'll be important. And this actually extends previous finding that we had, that lactobacilli are found in greater abundance in infants of mothers with lower levels of anxiety. And so we really do think that some of the changes we're seeing in mice are going to prove to be true in humans as well, which gives us a real opportunity to intervene, which I'll get to in a moment. And so I just couldn't believe that something that we're seeing in our rodent model actually turned out to be relevant in a human model. And so for any physician scientists, you can imagine my delight at that. And I'll keep moving in the interest of time. So this is to summarize the part of the talk that I've just given you, which is that there was an increase in natively vaginal taxa and opportunistic pathogens from late pregnancy to parturition. Higher levels of stress and depression were associated with increased CCL2 in the mother in the third trimester. And there actually was a relationship between a beneficial commensal microbe and a lower level of a beneficial microbe and a higher level of CCL2 in the umbilical cord at delivery. So I really do think that we're onto something. For the last part of my talk here, I'm going to focus on microbial metabolites. So these are bioactive metabolites that the microbes are producing, and we think that this is really key in transmitting stress from the mother to the next generation. So tryptophan is gotten a bad rap, both a good and a bad rap in terms of its ability to modulate social behavior. So there's been clinical trials where individuals with autism spectrum disorder have been treated with tryptophan to try to improve their social behaviors. And there's been some successful trials as well as some negative trials that don't show any benefit. And I really think that this is something worth thinking about more globally, is that if stress is reducing tryptophan metabolizers, which I'm about to show you, and you treat someone with tryptophan, if the microbe isn't there to metabolize it, could that be explaining why there isn't a benefit seen? Because the truth is both the host, and we're the host in this scenario, and microbes can both metabolize tryptophan. It's just that the host tends to metabolize tryptophan into more neurotoxic or less beneficial metabolites like quinolinic acid, whereas microbes tend to metabolize tryptophan into more beneficial and anti-inflammatory metabolites. And so really what the hypothesis that my lab has started to investigate is whether or not prenatal stress influences neurodevelopment through this dysregulation, through affecting this balance of tryptophan metabolism. So the tryptophan story is that, again, tryptophan comes from the diet and is taken up by enterocytes in the gut, or the host, or metabolized by gut microbes. And these are metabolites that should be familiar to all of you, including serotonin, but kynurenine has a really important role in immune function. Indole is very anti-inflammatory, and higher levels of indole is associated with better health. So we really wanted to start asking this question about is this part of why neurodevelopment is impacted by prenatal stress? And so the first thing we did was look at tryptophan in the contents of the dam. So we took fecal samples from there, and we did find a significant increase in tryptophan, suggesting that there's the tryptophan metabolizers were reduced. And then we've now shown this in 4 or 5 cohorts of mice. This is one of our most replicable findings. So I really believe it is that Parasitarella excrement hominis, try saying that 5 times fast. I can do it, but I'll do it later if someone asks, is significantly reduced in the dams with stress. So again, a really key tryptophan metabolizer in the mom is significantly reduced with stress. And this continues on into in the offspring. Again, this idea of bequeathing or setting up the gut-brain axis. So this is now into 5 weeks of age when we compared stress to control mice. Parasitarella was reduced in the offspring, and Bifidobacterium, another very key tryptophan metabolizer, is also reduced in the offspring of exposed to stress when compared to non-stress controls. And what was even more surprising to me was that this translates as well. So this is a collaboration with Chris Dunkel-Shutter and her collaborator, Mary Cousins-Reed. They did a really wonderful job phenotyping and defining the moms, and then we were lucky that they shipped us the samples from their infants. So the OASIS is a scale of anxiety, and what we found in our analysis from these infants, from these human infants at 6 months, was that if infants exposed to higher levels of anxiety had a significant, during pregnancy, in both the first trimester and the third trimester, had a significant reduction in this very key tryptophan metabolizer, Bifidobacteria dentum. And so it's changed in mice, it's changed in humans. What we then wanted to ask is this what's responsible for some of the changes I've shown you now in microglia? Remember, they have increased inflammation. They're important in neurodevelopment. I've shown you increases in psychiatric disorder susceptibility genes. So could this be due to changes in these tryptophan metabolizers? And how do we think that's working? It's through the AHR. The AHR receptor in the brain, it's found on microglia, it's found on neurons, astrocytes. We found a significant increase in the AHR receptor in mice exposed to stress. So that's how we hypothesize. So there's a lot of work to be done in this space that we're currently doing, that this could be a potential mechanism. So how do we do that? Well, if something goes down and we think that's bad, pretty basic neuropharmacology. You give it back and see what happens. So we did that throughout the stressor in the mom. We gave back Bifidobacteria dentum or Parasitarella excrement hominis here to simplify it as probiotics. We did fetal brain analyses. We also analyzed the brain and behavior in adulthood. And what we found, first I'm showing you here is data from the dam, is that we found that treatment here for the purpose of this slide, I'm showing you bifidobacteria dentum. One of the most significant findings we have is that the dams exposed to stress do not gain weight appropriately throughout pregnancy. And we actually found, and that's not due to litter, that's even after we normalize for litter size. So you can see that here in black, that's a significant reduction in weight. Bifidobacteria dentum actually restored their weight gain, which was nothing we've ever done has restored their weight gain to normal levels. It also significantly reduced the levels of CCL2 in the maternal plasma. And then it increased, the bifidobacteria dentum decreased IL-6 in the fetal brain, and then reversed this reduction in social behaviors in adulthood. The treatment here did. You can see this was with stress, it was reduced again, and then the treatment with a probiotic returned this to normal levels, and this is female offspring. So by giving back this beneficial commensal microbe, we actually did see a reversal of some of these findings that I've been showing you throughout my talk today. So we're very excited about this and hard at work following this up. So the conclusions is that I didn't waste a decade of my life, but the real conclusion is that prenatal stress is associated with altered microbiome in pregnancy, in both human and mouse pregnancy. CCL2 is uniquely influenced by stress in the microbiome, associated with microbiome in both preclinical and now a clinical study as well. And I really do believe that tryptophan and other microbes are really a very tantalizing translational target for intervention to help both the mother as well as the developing offspring. And with that, 11:58. I'm very pleased not to have run over. I hate running over. I'm going to thank members of my lab who did a lot of the work that I showed you today and are very hard workers. I'd like to thank my husband for collecting all those samples, and then my mentor, Mike Bailey at Nationwide Children's, as well as all of my funding sources, and all of you for listening to me today. And with that, I'm going to hit escape and stop sharing so that I can finally see all of you. I've been